Ladies and gentlemen, may I please have your attention as we are about to begin. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning and best wishes to us all. I would like to say welcome to Director of Research Center for Bioteknologi, Indonesian Institute of Science, Dr. Puspita Lisdianti, our Honorable guest speaker, Professor Satya Sugahara, PhD, from Graduate School of Agriculture, Ehime University, Japan. Our moderator for today, Dr. Edi Naroputra, and all participants this morning. First of all, let's be grateful to Allah Subhanahu wa Ta'ala, who has given us good health and time so we can attend and participate in the knowledge sharing seminar today. Next, we would like to invite the Director of Research Center for Bioteknologi, LIPI, to give welcome speech and open this knowledge sharing seminar. For Dr. Puspita Lisdianti, time is yours.
Thank you very much, Mbak Wardah, for the warm introduction. Okay. Assalamualaikum warahmatullahi wabarakatuh. Good morning, Ohayo gozaimasu, all of the participants. And also thank you very much, Professor Sugahara, that you can accept our invitation to give to share your knowledge. I think in the food biotechnology, related to the tea. Actually, every week we do the knowledge sharing seminar, and then researchers in not only from Indonesian Institute of Sciences, but also from Indonesia can join this webinar. Today, we are lucky because we will learn about I think from me, I'm a microbiologist. From me, the important thing is I would like to learn is about the functionality, how to identify the function of the food. Yeah, I think it's very important. And then for the elucidation of maybe the structure, and then also the application of the bioactive to the some anti-allergy. I think I would like to learn from Professor Sugawara. Okay, let's start for the knowledge sharing. Yeah, thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for Dr. Puspita Lisdianti. Ladies and gentlemen, before we begin the main session, let us start by saying a prayer first so that even today will run smoothly without any obstacle. Prayer start. Prayer finish. For the next session, we would like to invite Dr. Naroputra, the moderator for this session, please.
Thank you, Bu Wardah. Thank you, Dr. Lilis. And also, thank you, Prof. Sugahara. So, good morning, everybody. Welcome to this knowledge sharing seminar session held by the Research Center for Biotechnology, Indonesian Institute of Sciences. As mentioned before by our director, Ibu Lilis, that the KSS, or knowledge sharing seminar, is held by the Research Center for Biotechnology once a week, especially in online, in pandemic situations, we conduct it online, yeah, to leverage understandings and also a knowledge of the Indonesian scientists, Indonesian academicians, students, and so on, by inviting professors, practitioners, or researchers from Indonesia and also from overseas. So I am Naro. I will be serving as your moderator in these 4D sessions. Before that, I would like let me share a screen, yeah, to introduce Professor Sugahara background. So, Bapak Ibu, as you can see here, the full name is Professor Takuya Sugahara. He was born in Japan, I think in the city of Fukuoka, Sensei, Kyushu, I don't know. Yeah. On February 12, 1966. He graduated from the Faculty of Agriculture, Kyushu University, and Graduate School of Agriculture for his bachelor's, master's, and finally he received PhD in 1993. He has several work experience started from the assistant professor at Ehime University and becoming a research associate at the University of Texas Health Center in the United States, associate professor at the Faculty of Agriculture at Ehime University, and in 2013 he was granted a full professor title by the professor committee from the Faculty of Agriculture at University. From 2013, act as a director of Food and Health Sciences Research Center, and from 2016 until now, he was appointed by the president of the Ehime University to be the special aide to the president. He did and do research in food science related area, especially in functional molecule science and animal cell technology. He has been publishing 160 international academic papers and been recognized by Scopus with H index in 23. So here are latest or recent publications from Professor Sugahara. We may see quite a broad spectrum of the research area, from the stimulations of IgM production in the study of the immunostimulatory activity by egg yolk extract from Indonesian native chicken egg, together with Professor Armayani and friends, and also studying about the anti-allergy properties, yeah, by observing anti-degranulation properties, and then anti-allergic activity, and so on and so on. And also he was conducting research to authentify the coffee products by using SNPs, single nucleotide polymorphisms genotyping. And also he was I think he is still conducting research on cancer or anti-cancer activity from herbs, spices, or any food-derived substances. I think that's all introductions, short introductions about Professor Sugahara. And for the audience, let's take a lesson or learn from what Professor Sugahara delivered today, and hopefully it could inspire you. And if you got inspired by the talk, you may continue to the personal discussions, in order to build collaborations between your institutions or you yourself with Professor Sugahara. And if you have any questions, you may type in the chat box, and we can start the discussion sessions after Professor Sugahara delivered all the presentations. So for the next 45 minutes, time is yours, Professor Sugahara.
Oh, thanks so much for the nice introduction, Dr. Abhinav. So I will share today's presentation. Maybe you can see my presentation. And how about the sound? Is it no problem? It's clear? It's okay?
It's okay, Sensei. We can see it clearly.
Okay, thank you. So I will start my presentation. It's my honor it's my honor I can present my study on anti-allergy potentials of fermented black tea. So fermented black tea means the fermented tea leaf fermented by the fungi and lactic acid bacteria. So this is very unique tea in the world. So in Ehime, I am living in Ehime. So Ehime is very famous in the black tea. So there are one specific black tea in Ehime. So in my laboratory, we are evaluating the health function of fermented tea. So we found the anti-allergy potentials of the fermented black tea. So I would like to introduce the effect of the fermented black tea on allergy responses. At first, I would like to introduce myself. So I'm in Japan, Matsuyama, Ehime. This is the map of the Japan. So the Ehime is in the Shikoku Island. Shikoku Island is one of the major 4 islands of Japan. So, but it's a smallest island. So here is the Shikoku Island. So there are 4 prefectures. Ehime is here. Here is Ehime Prefecture. Then the capital of Ehime is Matsuyama. I'm in Matsuyama, and Ehime University are located in Matsuyama City. So Matsuyama is a local area in Japan. So the west side of Japan. So it takes one hour from Tokyo by a plane. So it's a little bit local areas. So the Matsuyama city is very beautiful city. So there is a castle, old castle. It was built maybe 400 years ago. So Matsuyama Castle is a very beautiful architecture. So the Matsuyama city is very famous in hot spring. The Dogo hot spring is very famous in Japan. So it's a wooden-made buildings, and then the very old one. So it's also built in. It was also built in maybe 400 years ago. A very old one. So this is the scenery from the hillside of Matsuyama Castle. It's a city. The population of Matsuyama city is 500,000. 500,000 people live in Matsuyama city. Not so big city. Not so big city compared with Tokyo, Osaka, Nagoya, Fukuoka. But very big. It's a small city, but very good weather, and it's good for living. So it's autumn festival. Maybe there are so many people have this is the Mikoshi in front of the Dogo Onsen, from Dogo Hot Spring, in front of Dogo Hot Springs. So this is the autumn festivals. It's very famous. So this slide shows the Ehime University. This picture was taken in spring season. So this is the cherry blossom who blooms, the cherry blossoms, very, very, very beautiful flower. So it's the main gate of the Ehime University main campus. This here is the main campus. Here, this building is the library, Ehime University libraries. So I'm in the studying in the Faculty of Agriculture. So Faculty of Agriculture is in the different campus. It takes 10 or 15 minutes by car from main campus. Here is the main building of the Faculty of Agriculture. And this is the main gate. So the Ehime University, we are developing human resources who are active in the many fields. This is the catchphrase of the Faculty of Agriculture. So in my study field, I am evaluating the food health function of many kinds of food. Especially Ehime is very famous in citrus. So citrus also containing the many kinds of health functional substances, such as the flavonoids and carotenoids and so on. It have the anti-allergy effect, especially nobiletin is a polymethoxyflavonoids. It has the anti-allergy effect. So we are developing the some functional food using citrus peel because nobiletin is contained in citrus peel. So we are making the yogurt containing the citrus peel. This is the citrus peel powder, and there's a jelly, and it's a tablet. So many kinds of tablet. This one is a newly developed tea. It's just a tea made with the citrus leaf. It's a fermented citrus leaf. So it also containing the many kinds of flavonoids. So it also have the some functions. So in my laboratory in Ehime University is collaborating with the many Indonesian universities. And we are evaluating the many kinds of Indonesian food stuff. Arrowroot, bangkwang, these also have the immunostimulatory activities and anti-allergy activities. Snake fruits, kedren, porang, red fruits, it's the products, and the ongkong, hakoi. This we are evaluating these many kinds of Indonesian food stuff and some processed food. So we are cooperating with the many Indonesian universities, UGM, University of Papua, and Jember, and ICR. So with these universities, we are evaluating some food functions of Indonesian food stuff, immunostimulating, anti-allergy, anti-inflammation, anti-obesity, etc. So by conducting this kind of studies, we are publishing the papers with Indonesian researchers. So our study contributing the SDGs, especially number 3, good health and well-being, and developing industrial innovations and collaborations. So we are moving to the black tea. We're talking about the, I will talk about the tea classifications. So the unfermented tea is a green tea. Japanese green tea is unfermented tea. Yield tea leaf will steam to inactivate the internal enzymes. And then it's a very fresh green tea. And then green tea is very, very popular in Japan. So the half fermented tea is, the next one is the half fermented tea. The oolong tea in China is categorized in this half fermented tea. It's fermented by the internal enzymes. And fully fermented tea is the black tea, or this fermented tea also fermented by the internal enzymes. So today I would like to introduce the fermented black tea, the microorganism fermented tea. It is a very special fermented tea. The tea leaf is fermented by the microorganisms, by lactic acid bacteria or fungi. So the not internal enzyme, but the microorganism fermented. So the Arabic fermentation, under the Arabic fermentation, it's a bata cha. It's produced in Japan, Toyama Prefecture. And anaerobic fermentation, it's our bancha tea. It is produced in Tokushima Prefecture in Shikoku Island. And this is a two-step fermentation. The first step is aerobic fermentation, and the second fermentation were conducted under the anaerobic fermented situations. So this two-step fermentation process was conducted to produce Goishicha tea and Ishizuchi black tea. Today, my study focus were on the Ishizuchi black tea. So these two tea is these two tea are fermented by under the aerobic fermentation, the fungi fermentation, and the second step on aerobic fermentation process, the lactic acid bacteria are conducted. Lactic acid bacteria were used for the second step fermentation. So this is the very special, very unique tea in the world. Maybe you know the miam in Thailand is also fermented tea. Maybe you are familiar with the maybe you know the miam in Thailand is also microorganism fermented tea. So in Japan, these four kinds of fermented tea, there are four kinds of fermented tea in Japan. So the Ishizuchi black tea is produced in the Ehime Prefecture. Two-step fermented black tea, they fermented by fungi and lactic acid bacteria, lactobacillus. And the other one is the other two-step fermented tea is Goishi black tea, Goishicha tea, produced in Kochi. Kochi also in the Shikoku Island. A two-step fermented tea fermented by fermented process of goishi tea is almost same as Ishizuchi black tea. So, as well as Ishizuchi black tea, goishi cha tea also fermented by two steps, aerobic and anaerobic situation conditions, under the aerobic and anaerobic conditions. So bata tea, it's produced in Toyama Prefecture. So the tea leaf are fermented by fungi, only fungi, under the aerobic conditions. Bata tea. The final tea is the our bancha tea. Our bancha tea is also fermented microorganism fermented tea. Fermented by lactobacillus under the anaerobic conditions. So 3 or 4 fermented tea in Japan are produced in Shikoku Island. So this is Shikoku Island. So here is Matsuyama, capital of Ehime Prefecture. So the Ishizuchi black tea are produced and this area. And goishi tea in Kochi are produced in around here in the mountainside. And our bancha tea in Tokushima is produced around here. These 3, 4 fermented tea, 3 or 4 fermented tea in Japan are produced only these areas. So the microorganism fermented tea in Japan is very specific in Shikoku Island. So this is the Mount Ishizuchi. It's the highest mountain in west side of Japan, almost 2,000 meters high. So under the hillside of the Ishizuchi, Mount Ishizuchi, the 3 fermented teas are produced, and in this hillside of Mount Ishizuchi. So there are maybe I guess that there are some specific microorganisms good for fermentation of tea leaf. I just guessing. I'm just guessing. There is a very good area for the producing fermented tea. So in this slide showed how to make Ishizuchi black tea, the process of the tea leaf, processing of tea leaf. At first, obtained and yield tea leaf are steamed for 1 hour. It is steamed like green tea. The internal enzymes are inactivated, and unwanted bacteria also inactivated by steaming process for 1 hour. After steaming, the first fermentation, primary fermentation will be conducted under the aerobic conditions. Fermentation by aspergillus, one kind of fungi, for 1 week in a wooden-made box. In the aerobic fermentation by aspergillus, like this. It takes 1 week. And after fermentation by fungi, aspergillus, tea leaf crumpled will be crumpled like this. Just tea leaves are crumpled. after crumpled, the final step is the secondary fermentation under the anaerobic conditions. In this second fermentation, the tea leaf are fermented by lactobacillus, mainly lactobacillus plantarum, the main bacteria for in this fermentation process activated conducted by the lactobacillus plantarum for 2 weeks. So the one step, primary step, and the secondary step, aerobic and anaerobic conditions, fungi and lactic acid bacteria, the fermentation process of tea leaf conducted. And then finally, the tea leaf are dried under the sun, naturally dried under the sunshine. And then the tea leaf will be made. So this fermentation process by fungi and bacteria, lactic acid bacteria, the concentration of the catechin, EGCG, epigallocatechin gallate, EGCG is a major component in tea leaf, Japanese tea leaf. The catechin contains in Ishizuchi, IBT means the Ishizuchi black tea. So the EGCG is a major component in tea leaf. So EGCG also have the anti-allergy activity, very famous in Japan. So this is the analysis of the EGCG standard. So this peak is show the EGCG. So the Ishizuchi black tea fermented by bacteria. So EGCG are metabolized. So the EGCG contents is almost zero. No concentration. Almost all EGCG are metabolized by the bacteria during fermentation. So the EGCG is a major component in tea leaf. However, EGCG concentration are decreased during fermentation, as indicated in this chart. No peak of EGCG. This figure shows the internal addition of the EGCG in the IBT. So this peak is the EGCG. So compare with these two figures, no EGCG in EGCG black tea. So this is basic information of the functional substances in EGCG black tea leaf. So no EGCG in EGCG black tea. Okay. So this is the concentration content of the EGCG black tea before and after fermentation. EGCG concentration almost zero. Epigallocatechin gallate, ECG also almost zero. So the totally catechin concentration less than 1/10 compared with the green tea before fermentation. So the many of catechin components are metabolized by during fermentation. So the content of catechins are 1/10 before fermentation. So we move to the anti-degradation effect of acidity black tea. So the function of fermented tea. So this slide shows the preparation of IBT samples. Leaf of acidity black tea are suspended in distilled water. Tea powder, tea leaf are suspended in the water, and then extracted at very high temperature, because we want to retrieve the maximize the retrieval of the components in tea leaf. So we use the very high temperatures. After extraction, the insoluble materials are removed by centrifugation, and then correction of supernatant. The supernatant was dialyzed by molecular weight cutoff 14 kilodalton membrane against distilled water to eliminate the lower molecular weight components. So by dialysis using 14 kilodalton molecular cutoff membrane, we can eliminate the low molecular weight components. So so we use the high molecular weight only. We obtain the high molecular weight components, only high molecular weight components in acidic black tea. Then after dialysis, filtration for sterilization. And then we use this sample as IBT extract for assay. So this is the scheme. This shows the type 1 allergy. Summarize the type 1 allergy. So base of one kind of white blood cell, basophils and the mast cells participate in the type 1 allergy. So in basophil or mast cells, there are granules containing histamine. It have the many granules in cytosol, and this one is the Ig receptor. There are cell surface Ig receptors on basophils and mast cells. So IgE in blood bind to the Ig receptor on basophils or mast cells, like this. So and IgE bind with the antigen. It induced the allergy. I mean, we call it allergen. This allergen, it means the antigen induced the allergy response. So binding of allergen on mast cells via IgE, this binding induced the signal transduction to release the granules. So the antigen stimulation, the granule will be released from cytosol and the blood, and the histamine also released. The released histamine stimulate the epithelial cells to induce the allergic response, responses. So this release of the granule from cytosol or basophil or mast cell to the blood means the degranulation. So this process, release of the granules from cytosol to the blood, it means the degranulation. So in our experiment, we check the anti-degeneration activity. We evaluate the anti-degeneration activity of IBT. Was evaluated. So we use the RBL2H2 cells. This is a basophil cells derived from rat. This is a cell line, RBL2H3 cells obtained. This is the rat basophilic cell line. We use these cells for evaluation of anti-degeneration activity of IBT. So addition of the IBT clearly suppress the degeneration in a dose-dependent manner, like this. At the highest concentration, degeneration was significantly suppressed. So this means the IBT. This also show the same data after fermentation. IBT have the anti-degeneration activity, like as shown in red lines, red symbols. Very high, very strong anti-degeneration activity. As I mentioned before, green tea containing catechins. Catechins also have the anti-degeneration activity. So we compared with the anti-degeneration activity of IBT with green tea. Green tea means before fermentation. Before fermentation, green tea containing the catechins, so the catechin can suppress degeneration like this. However, I already introduced IBT does not contain the catechins. However, very strong anti-degeneration activity compared with green tea. A specific activity was significantly increased. This means the IBT does not contain the catechin, but it has the anti-degeneration activity. This means the active substance in IBT is not catechins, but it has some novel functional substances in IBT. It has the anti-degeneration activities. So this slide also show the fermentation by bacteria is very useful process to produce anti-degeneration, anti-allergy substances. So this shows the fermentation is very important process. Bacterial fermentation is very important process to produce anti-degeneration substances in tea leaf. So we examine the molecular weight of the active substance in IBT. As I mentioned, IBT was diarized with molecular cutoff of 14 kilodalton. So it containing the relatively high molecular weight substances. So we check the activity, molecular weight, and of the active substance in IBT using Gale filtration method of liquid chromatography. Molecular weight of the active substance in IBT was estimated by Gale filtration. We conduct charge the IBT to the Gale filtration to evaluate the molecular weight. So the very clear peak was observed, and in the 12.91 milliliter, very clear sharp peak. So we estimate the molecular weight of the peak substances using standard curve. So the molecular weight of the substance in IBT was estimated as 24,000 Dalton. So the active substance in IBT, molecular weight of the active substance in IBT was estimated at around 24,000. So to identify the active substance in IBT, we fractionated the IBT using liquid fractionation process. IBT was at first step. IBT was mixed with ethyl acetate. So the water layer and the ethyl acetate layer was obtained. However, ethyl acetate layer, this layer mainly containing the thiaflavin. Thiaflavin is a low molecular weight substance in fermented tea, but no activity, no anti-degranation activity. So the obtained water layer are further fractionated by normal butanol and water layer and the normal butanol layer. So the normal butanol layer containing the thiarubidin. Thiarubidin also polymer of the flavonoids, polymer of the polyphenols in fermented tea. So it had the thiarubidin fraction showed the anti-degranation activity low, very weak anti-degranation activity, only 23% suppression. So they obtained water layer after normal butanol fractionation. So we add ethanol. So water layer and precipitate were separated. So the precipitates are mainly containing thiar-brown. Thiar-brown is the polymer of polyphenols. So low, high molecular weight polymers or polyphenols, thiar-brown. The color of the fermented teas come from thiar-brown. Thiar-brown is the color of the thiar-brown is very dark color, almost black color. So the color of IBT is mainly come from the thiar-brown substances. So we evaluate the anti-degranation activity of thiar-brown fraction. The fraction suppressed almost 70% of degradation. So this fractionation process, to conduct this fractionation process, we can identify the active substance in IBT is mainly thiar-brown. So thiar-brown, as I mentioned, thiar-brown is the polymer of polyphenols produced during fermentation. The thiar-brown is not contained in fresh tea leaf. So the thiar-brown is produced during fermentation by bacteria, fungi, and mainly lactic acid bacteria, Lactobacillus plantarum. So from this figure, we can identify the active substance in IBT is thiar-brown. Mainly thiar-brown. Some weak activity was observed thiarubidin fraction. So thiarubidin also the active substance. However, major one is thiar-brown. produced during fermentation. So this slide also shows the fermentation process is very important. So the comparative investigation of the RBT, TB. So how fermented tea? The example is oolong tea. Oolong tea is the half fermented tea, and microorganism fermented tea, puer tea fermented by lactic acid bacteria. So the two-step fermented black tea is black tea. We obtained Tier Brown fractions of oolong tea, puer tea, and the is black tea, and compared the anti-degradation activity. Oolong tea, puer tea, is black tea, Tier Brown fractions. As a result of the experiment, no activity observed in oolong tea. Oolong tea is the half fermented tea, however, not bacteria, but internal enzyme. So the oolong tea, Tier Brown from oolong tea is no activity to suppress the degradation. However, puer tea fermented by lactic acid bacteria and IBT have the Tier Brown fraction of puer tea, and IBT have the anti-degradation activities. So the fermentation by bacteria is very important, especially maybe lactic acid bacteria fermentation is very important to produce the active substance in fermented tea leaf. So we need to evaluate the mode of action of IBT, how IBT suppress the degradation. So we're using Western blotting analysis method to evaluate how IBT suppress the signal transduction under the Ig receptor, Fc-R1. So under the Fc-R1, Sic, ring, cascade, there are Sic cascade and the ring cascades, like this. Sic was phosphorylated, and then phosphorylated Sic activate the PI3 kinase or PLC gamma 1 and the PLC gamma 2 by phosphorylation. And then finally, calcium ion concentration in cytosol will be increased, and then degradation induced. This is the Sic pathways. So in the Sic pathways, the phosphorylation of Sic was significantly suppressed by IBT, like this. So the phosphorylation of Sic was suppressed. Then finally, PLC gamma 1 and phosphorylations of PLC gamma 1 and gamma 2 also suppressed, because this is the downstream of the Sic. So the phosphorylation of Sic was suppressed, then PLC gamma 1 to activation were suppressed. And the PI3 kinase, activation of PI3 kinase also suppressed by IBT, like this. So from this result, IBT suppressed the signal transduction, degradation signal transduction at Sic. Then suppression of SYK induced the downregulation of the activation of PLC gamma 1, gamma 2, and PLC kinase, and finally suppress the increase in calcium ion concentration, then downregulate the degranulation. So next step, we evaluate the anti-cedar-poenosis activity of IBT in vivo. So experimental animal, we use the biopsy mice, and this is the CRY-J1 is the cedar pollen allergen. So you induce the poenosis in mice by injection of the CRY-J1. And we administer the IBT instead of water. It's a free access. Then finally, we check the allergic symptoms. Labing action was significantly suppressed by the control. Control means the administered just water. However, IBT administration significantly suppress the labing action of poenosis. So the intake of IBTs isn't the poenosis symptom in vivo. So the other anti-allergy activity of IBT was evaluated. The next step is the IgE production suppressing activity of IBT was evaluated. So our type of allergy response, at first step, we evaluate anti-degranulation activity using basal saline. So the IBT tear brown fraction can suppress the degranulation. So the previous step of type of allergy, release of IgE. IgE is a major antibody which induce type of allergy. So suppression of the IgE production in body maybe ease the allergic symptoms because the IgE is a major antibody. IgE is the antibody which induce type of allergy. So the suppression of IgE production is a key step for the suppression of the allergic reactions. So we evaluate the effect of IBT on IgE production. So the suppression of IgE level is very important step to ease the allergic symptom. So we use the IgE producing animal cells. We use human leukocyte cell line U266 cells, which producing IgE. So we add the IBT to the culture media of U266 cells. So the addition of IBT is significantly suppress the IgE production. So this is the dose response, and this figure shows the time course of the effect of IBT. So after 12 hours of addition of IBT, the production levels clearly suppressed. And the next 12 hours and the next 24 hours, the accumulation of IgE was completely suppressed like this. So the IBT can suppress the IgE production by U266 cells. So we evaluate how IBT suppress the IgE production. We focus on the messenger RNA expression level. So many normally the production of protein production, when the protein production was suppressed, in many cases, the messenger RNA level of coding the protein will be suppressed. However, the IBT clearly suppress the Ig protein production level, as shown in this figure. However, no effect on IgE messenger RNA expression level, no effect on suppression of Ig messenger RNA. So in normal case, the suppression was suppressed when the protein production level was suppressed. Messenger RNA level was also decreased, suppressed. However, IBT did not suppress messenger RNA level, even though the IBT suppressed the Ig level. So this means the IBT suppressed the transcription level. IBT did not suppress transcription level to suppress the Ig protein. So we speculate we need to speculate how IBT suppresses the Ig production. So we focus on the translational process. We need to further experiment on the mode of action of IBT. So
another 10 minutes.
Okay. So identification of the active substance in IBT. So the IBT was treated with the trypsin and the proteinase K. However, no effect on activity of IBT. This protease treatment were not affect on the this two protease treatment did not affect on IBT activity. So we retrieve the tear brown fraction of IBT. Then we check the Ig production effect on Ig production. So tear brown fraction clearly suppressed the Ig production level compared with the IBT. So the specific activity was increased by using tear brown fraction. So in this case, tear brown is the key factor of decreased Ig production. So the next step, we evaluate the effect of IBT on serum Ig level in vivo. So allergy was induced by overalbumin, this OVA from chicken egg. So they administered the OVA to induce the allergy. After allergy induction, during allergy induction, we administered IBT by free access to IBT instead of water. So after OVA induction, allergy induction, we collect the blood to evaluate the immunoglobulin level in blood. So Ig level was clearly significantly decreased by IBT administration. However, other Ig class, IgM, IgG, IgA were not affected by IBT administration. So specifically, Ig level was clearly suppressed by IBT. So this means the anti-allergy, anti-IgG level activity was observed in vivo. So the conclusion, ECG black tea suppressed the degradation of basophilic saline RBT cells. IBT suppressed the phosphorylation of SYK in RBT2H3 cells to suppress the degradation. This is we can show the mode of action of IBT to suppress degradation. So the IBT suppress isn't the cellular paralysis symptom in vivo. So IBT also suppress not only degradation, but also Ig level, suppress Ig level in allergy model mice, and also suppress the human U26 cells producing IgE can be suppressed by IBT. So this is the final slides. The basophilic saline cells, the granule are released by the antigen. So the IBT can suppress type 1 allergy by 2 steps. IBT suppressed Ig production level. Ig production level was suppressed by IBT. And IBT also suppressed the degradation. So during this type of allergy process, IBT can suppress 2 steps. One step is the Ig production step, and the second one is the degradation step. So IBT suppressed the Ig production level by B cells, Ig producing B cells, and IBT also suppressed degradation of basophilic cell lines, basophilic or mast cells. So this we can show the IBT is very effectively easing the type 1 allergy symptoms by Ig level decrease by down regulation of the degradation. So the fermented black tea is a very useful for the anti-allergy functional food staff. So thank you for your kind attention. Thank you so much.
Thank you so much for the presentations and then the talk, Sensei. I think there are several important things to recall in our memory what Professor Sugahara has been talking about. The first, that the fermented tea is really unique, especially in the Shikoku area where the university located in. And also, Professor Sugahara delivered some information regarding the functionality of the fermented black tea, especially the ECG black tea, that has been evaluated in several methods, in vitro methods, Western blotting analysis, and also in vivo assay or in vivo methods. So from the talk, we learned that ECG black tea lost the epigallocatechin gallate, or catechins, because of the fermentation process. However, the degradations of the catechin is expected or directly or non-directly related to the productions of a new compound that can be identified by the research done by Professor Sugahara and team that we know during the presentation is called as Thea Brown. So we can also see that in vivo assay, the inductions of the allergen in mice, the allergy inductions by allergen in mice is suppressed or was suppressed by the treatment of the theobromine to the mice itself. So, ladies and gentlemen, we are now entering the discussion sessions, question and answers. You may raise your hands and ask to Professor Sugihara directly, or you may type your questions in the chat box. You may also type your questions in Bahasa Indonesia, then I will try, or our team will try to translate it to English. But before that, let me deliver two questions, Sensei. Two questions that has been asked to me directly through the chat box. The first question is, you said that the fermentation of Isizuchi black tea, the fermentations of tea by lactic acid bacteria will will produce will increase the activity, the anti-allergic activity, right? Any possible explanations on how this lactic acid bacteria produce the active substance? And then the second about what lactic acid bacteria is good to use.
Thank you. So I'm sorry, I cannot follow your questions. So can you repeat it?
I will deliver it one by one. The first
question,
The first question: do you know or do you have any thought on how the lactic acid bacteria can produce the active substance which can induce which can have anti-allergy activity? On how the lactic acid bacteria produce the active
substance. How to metabolize the component in the tea leaf?
Maybe the questions like that.
Maybe. Yeah, unfortunately, we cannot identify how the lactic acid bacteria makes tea brown. So of course, it's during the metabolism of the lactic acid bacteria. So, but we did not identify the metabolic pathways. Unfortunately, we did not identify the metabolic pathway to produce the tea brown.
Okay, Sensei. The second question: any specific lactic acid bacteria instead of just Lactobacillus plantarum that can produce the active substance? Thank you.
Yeah. So we identified the clone the lactic acid bacteria and the fungi from the tea leaf of the Isizuchi black tea. So we can identify the Lactobacillus plantarum. Lactobacillus plantarum is a major lactic acid bacteria in some food and vegetable foods. So maybe the tea leaf also have the plantarum on the tea leaf. So so because the plantarum is a major bacteria, lactic acid bacteria in the plant leaf or something. So so it easily contribute to the fermentation of the tea leaf. So and the fungi. So also we can identify the aspergillus and the fungi on tea leaf. So the aspergillus also on tea leaf contribute to the first step fermentation, and the second one is the major bacteria as a major bacteria. Lactobacillus plantarum is contribute to the second process. So the maybe some minor lactic acid bacteria are also contribute to the fermentation. Not only plantarum, but also some minor lactic acid bacteria are contributing for the fermentation, for the production of tea abalone. It's okay?
Okay, since I think it's clear, yeah. So I will give the questions. I will give the sessions to Mbak Sri Marlinda, ya, to ask questions. Please, Mbak.
Oke, terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you for this occasion. I have two questions for Mr. Takuya Sugaha. The first question is, does the side effect when someone has a degenerative illness, then they consume the IBTT?
Some other function? You mean some other function of the IBT?
Does the side effects, Mister?
Side effects, Sensei. Any side effects?
No. Especially we have not observed the side effect. Yeah, no bad effect on taking the ECG black tea. It also not only anti-allergy, but also IBT has the anti-obesity activities. It also has anti-obesity activities. So maybe I mean this is a two major function. So not side effect, but it has the anti-obesity activities.
Okay, thank you, Sensei. The second question is, what is the specific criteria for the genetic from the microbe that can use to be a fermented microbiome? What is the genetic criteria, Sensei?
Unfortunately, we did not evaluate the criteria of the bacteria, just the cloning and just the identification of the class of the bacteria. So I have not checked the genetic evaluation. Yeah. No, we have no information about the genetic one.
Okay, thank you, Sensei.
Thank you, Mbak Sri Marlinda. Are you using back sloping technique or are you using starter culture, Sensei, for fermentation?
No, just a normal, just a natural one. No addition of the clones bacteria, just a natural one. Not control. We did not control the concentration of bacteria and the species. It's just a natural one.
Okay. Another question from the chat box, and so we can read together in the chat box.
Yeah.
Maybe you may also stop the screen sharing. So I will read the questions from Dr. Dr. Nanik Rahmani. Thank you very much for your nice presentation. I would like to ask how to determine the concentrations of microbes to be induced into the fermentation process. That's the first question. How to determine the concentrations of microbes?
Actually, we did not determine the concentration. Just as I mentioned, it's just a natural fermentation. So we did not check. We do not check the species and the concentration of microbials. It's just natural one. Yeah.
I see.
Natural.
I think the rest of the questions from Dr. Nanik have been answered by Professor Sugahara. Yeah, Professor Sugahara did not focus on the evaluations of the microbes itself because the product is the product used was naturally fermented. Yeah, Sensei.
Yeah.
Okay. Bu Nani, if you have any questions, we welcome you to speak. Just unmute your microphone. Is it clear, Bu Nani?
Okay, thank you. Clear.
Thank you, Bu Nani. Okay, any other questions? Mbak Sri Marlinda, your hand is still raising. Do you have any questions? Oh no. Okay, I invite other questions. Sensei, another question asked privately to me to deliver it to you. How do we expect, Sensei? How do we expect that there is an active substance in food or in any food component, or maybe specifically how do we
speculate?
Yeah, speculate, something like that.
It's a very, very difficult question. We cannot speculate that, but we have information about some food stuff have some kind of effects and some. We can obtain the information about the epidemiological studies, maybe. Epidemiology studies conducted also in Indonesia. So the traditional food and traditional medicine is kind of this. It's a medicine, however, but it have many of traditional medicine from some food stuff. So our epidemiological study, we can obtain some information about the active substance in food stuff. So this is the reason why we focus on some spices and some herbs. It used as a traditional medicine, something like that. So as a result, we can identify some new components which have some health functions. So the information can be obtained from the traditional medicine, something like that. So maybe in Indonesia you have some traditional medicine or traditional food for which known as some health functions, right? It's a major source, informational source of to evaluate the functions. It's okay.
Okay, so we can use epidemiological studies or local wisdom, Sensei, in order to expect or speculate the active substance in foods. Okay. Okay. Sensei, another question. Do you have your own product that is now in the market based on your research?
Sorry, I can catch you.
Do you have your own product that is now in the market, Sensei, in which the product is developed by your team or your research?
Yeah. I'm sorry, your voice cannot catch because of the web conditions. So may have. Can you say again?
Okay, okay, Sensei. Maybe the problem is in my connection.
Hmm.
Do you have your own products? Your product? Do you have your own product? Your product that is now in the
Yeah.
market, Sensei? That is now sold in the market.
Yeah.
Indeed, the
Some
product is developed by your team or your research.
Yeah.
Can you show it?
There's some products.
I mean, I think in your slide you have mentioned that this person is not catching up before.
Some products, health function products. You mean that?
Yeah, like yogurt, something like that.
Okay. I was sure. I was sure. You mean this one?
Yeah.
So this all products are sold in market. So this yogurt and this tablet made from citrus pear tablet. It has the anti-allergy, and this one is the anti-inflammation. So so good for memories, and so this also have the containing the pear, citrus pear. It have the it's containing the some flavonoids. There is a cake, and it's it's ice cream also containing the citrus pear components. This made from the seaweed. Seaweed also have the many functional materials. This is seafood containing the some dietary fibers, and as I mentioned before, this is made from citrus leaf. Citrus leaf also containing the many kinds of flavonoids and carotenoids. So this tea leaf, citrus leaf tea also have some functions. You can take this tea, maybe you have even the allergic symptoms, something like that. Yeah, we made so the citrus is a very famous in Ehime, so we use the citrus, and especially pear is discarded. It's a byproduct of the squeeze to make the juice. So the citrus pear can be obtained after squeezing. So we reuse the pear powder to develop the functional food. Especially in citrus, some many kinds of functional materials are contained in not juice but pear. So we can use the pear for the functional materials for health, especially anti-allergy, anti-inflammation, anti-obesity. So it's a very good materials for developing the functional food. So this is the reason why we use the citrus pear for many kinds of products.
I see.
It's okay.
I see. Okay, Sensei. I think it's clear that you have been conducting research about citrus peel, and now it is in the market, right, Sensei?
Yeah, that's right. Yeah.
Okay. So I think the question is quite similar with Mbak Fitri questions, Mbak Fitri Setioningrum. The previous question is quite similar with yours. Any additional question, Mbak Fitri, or do you want to ask directly?
Yeah.
Mbak Fitri?
Clear, Mas Naro. Thank you, Sensei.
Yeah,
Okay,
thank you.
Thank you, Mbak Fitri. So another question from Mbak Fania. I think I will ask Fania to ask questions directly. Open your mic, Mbak Fania. Can you open your mic and ask the questions directly to Sensei?
Yeah, hello, Sarnaro, and hello, Sugihara Sensei. Thank you
Hello.
for the presentation. I would
Thank you.
like to ask regarding the fermentation days. What will happen if the anaerobic fermentation is faster than 2 weeks? Will the tea be made? Thank you.
So the aerobic fermentation process, the major bacteria is the lactic acid bacteria, right? So it takes 2 weeks. So it producing it also produces the lactic acid. So some acidic flavor, or can be you can feel the acidic flavor in the black tea. So during this fermentation process, the tea brown, the active substance in black tea is made during anaerobic fermentation, because the pure tea is made by only fermented by the lactic acid bacteria. Its tea brown fraction of pure tea also have the almost same activity compared with the black tea. So during anaerobic fermentation process, the tea brown, which is the active substance in fermented tea, is produced because maybe there's some, I don't know. As I mentioned, I don't know what kind of metabolism conducted in the bacteria, but during by the lactic acid bacteria fermentation, the active substances produced in tea leaf. It's okay.
Okay, Sensei. Thank you for the answer.
Thank you.
Thank you, Fania, for the questions. So I invite participants to ask questions. Any more questions?
Hello, Dr. Naro.
Oh, hello, Dr. Rosie.
Okay, may I give some question to Prof. Sugiyama Sensei, Sugihara Sensei?
Yes, please, Dr. Rosie, please. We are happy to hear.
Dr. Naro, and thank you very much, Sugihara Sensei, for your very comprehensive research and presentation. So my first question is, in beginning of the fermentation, you mentioned that the internal enzyme was also working during the fermentation. So could you please, what kind of the internal enzyme was working during the fermentation? And the second question, I don't know this is correlated to Dr. Abi question or not. Do you have some idea what kind of the specific activity from the Aspergillus or from Lactobacillus plantarum during the fermentation, whether they have produced some specific enzyme or specific metabolite, for example? Okay, maybe that's it, Dr. Naro. Thank you.
Okay, thank you, Dr. Rosie, for the questions. I think I may repeat for a moment for the first question, Sensei. You mentioned that there was internal enzymes involved in the first step of the fermentation, right? Can you give us clue what enzyme it was?
Yeah, we don't know. We do not speculate what kind of enzyme is needed for the fermentation for produce oolong tea or black tea. So we did not evaluate what kind of enzyme. However, we inactivate maybe oxidative enzyme is used needed for the tea fermentation, internal tea fermentation to produce oolong tea or red tea, something like that. So however, we did not identify the specific enzymes. However, it's categorized in the oxidative enzyme is very important for the fermentation. So I don't know what kind of enzyme in bacteria can induce to produce the TR-brown. Maybe it's also a kind of oxidative enzymes. Maybe it's kind of oxidative enzyme is contribute to the produce TR-brown. But we did not identify the enzymes in bacteria.
Okay, thank you for the answer. I think it's clear, yeah, Dr. Farozi, that the first questions cannot be answered by Professor Sugihara because it's quite out of the research scope of him. Hope you understand on this matter. And the second question, Sensei, any specific or unique substances that can be produced by Aspergillus or Lactobacillus plantarum instead of just the theobromine?
Yeah, actually, we have not precise examination about how they contribute these bacteria to produce the active substances. So actually, we have no information about the process for production of the theobromine by the Aspergillus and the Lactobacillus plantarums. Sorry.
Thank you for the answer. Is it okay, Pak Harurozi?
Okay, Dr. Abe. Thank you. Thank you, Sensei.
Thank you.
I think we may develop another collaboration. Is it possible?
Yeah.
For any of the participants here who are interested in developing or evaluating the mechanisms that or fermentation process that can scale up the concentrations of theobromine.
Yeah, of course. Yeah.
Okay, Bapak Ibu, you may continue the talk with Professor Sugihara because he was telling us in the beginning that he would like to accept Indonesian students, another Indonesian students to study at his lab for PhD program for maximum 2 students. Maybe you can discuss with him or discuss with me for future or for further information.
That's yours. Sorry, sir. How to join this research? Maybe we can use a scholarship max or another scholarship.
Sorry, who is speaking? I cannot see.
Sri Marlinda, sir.
Oh, okay, Mbak Sri Marlinda. Okay. How can they join? How can the participants become your students, your PhD students? So they should find the scholarship first, or
we have some scholarship programs provided by the mixed, so you can apply some PhD program in a human university. So the scholarship will be provided by the mixed. So, but it's a bit competitive, so it's a little bit difficult to be a scholarship student, but there is some chance. Yeah.
Okay.
Okay. It's
Thank you.
difficult, but it's possible for you, Mbak Linda. Yeah. So
Yeah,
please try to find out the information from the Mombu Show or Max website, and prepare the application form and prepare all the documents needed. And you may choose Ehime University, and you may choose Professor Sugara as your prospective professor in your proposal application.
Okay.
Yes,
Thank you,
Sensei.
sir.
Yeah.
Okay.
Thank you.
So
Thank you.
last questions. I invite last question from the participants. Any questions you would like to know about the research, collaborations, or how to say academic, yeah, PhD or masters? I invite you 1 or 2 questions. If no, so I will close the session. Any question from the person? Yes, Dr. Khairul Anam, please.
Thank you very
open
much,
your mic,
Mr.
Khairul Anam.
Naro. Thank you very much for your nice presentation, Sensei.
Thank you.
In this occasion, I would like to ask about maybe I missed from your presentation, but I would like to ask about the Thea Brown compound in the other tea products beside the IBT products, such as like anaerobic product, anaerobic fermentation tea only, and then also the aerobic fermentation only. Did you evaluate about their brown product in that kind of tea?
No, I just evaluate the Thea Brown from oolong tea. It's made from the fermentation by internal enzyme, and the other one is just yeah. Sorry.
You may share your screen.
That's right. So they just the oolong tea and the pu'er tea. So we checked only these 3 kinds of tea. So we did not evaluate the other kinds of tea, only fermented by anaerobic conditions, something like that. Yeah.
But in the aerobic fermentation, the response is almost similar with the IBT, right, Sensei?
Yeah, yeah.
Okay. Thank you very much, Sensei.
Thank you.
Thank you, Dr. Anam, for questions. Again, last question before I close the session. Last question. If no questions, I would like to say thank you very much to Professor Sugahara for the presentations and to share your knowledge to us, and also to open opportunity for the Indonesian scientists or academicians to study at your lab in the future amid this pandemic situations. I would also like to say thank you for Dr. Puspita Lisdianti, Dr. Lilis, our director, for staying from the beginning until the end with us. And also to thank all the participants to join the sessions. This is the session for us to take photo together. Or Ibu Lilis, you would like to say questions or say something? No, it's okay. Okay. Okay, so I will give the give the chance to Miss Dr. Ishana to lead us, or Dr. Dimas, to take picture together.
Okay, thank you, Mr. Naro. Ladies and gentlemen, this time I'm going to take the picture of all of us. So I'm asking you, Bapak Ibu, to activate your video. Bapak Ibu, dimohon kesediaannya untuk mengaktifkan videonya karena kita akan memasuki sesi foto bersama. So we have 67 participants in 3 slides, so I think it takes a few moments for me to taking a picture. So I'm gonna start now with the first slide. Please hold on. And then the second one. Yeah, and then the last one. Okay, thank you, Mas Naro.
Thank you, Mbak Isyana. So I will again thank you from me, the moderators. Apologize me for any mistakes I made. Thank you, Prof. Sugahara, and I will give it back to Ibu MC, Bu Warda. Please, Bu Warda.
Thank you for our moderator, Dr. Naro Putra. Ladies and gentlemen, we now come the end of our event today. Thank you for Dr. Puspita Lisyanti, Professor Takuya Sugahara, Dr. Naro Putra, and all participants. See you soon at the next knowledge sharing seminar. Thank you very much. Wabillahi taufiq wa hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you so much.
Thank you, Sensei.
Thank you. Thank you.
See you again.
Thank you so much. See you. See you. Thank you.
You may leave, Bapak Ibu. You may leave the sessions, the forum.